Greetings, everyone, and welcome to today's webinar, introducing the first dual reporter application for Luminex xMAP INTELLIFLEX DR-SE Instruments, measurement of polyphosphorylated and total proteins on the same bead. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. I'd now like to welcome our speakers. Dr. Sherry Dunbar, Senior Director, Scientific Affairs, Luminex, and Dr. Sigrun Badrnya, Manager, Research and Development, Protein and Cell Analysis, Thermo Fisher Scientific. Unfortunately, Dr. Dunbar was unable to join us live today. However, Dr. Badrnya will be taking live questions following her presentation. Now, we encourage you to submit any questions you may have at any point during the presentation, and to do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. We will now begin Dr. Dunbar's presentation.
Hi, I'm Sherry Dunbar, and I'm the Senior Director of Scientific Affairs Programs for Luminex and DiaSorin. I'm very happy to be with you today, presenting together with our partners at Thermo Fisher Scientific, introducing the first dual reporter application for Luminex xMAP INTELLIFLEX DR-SE Instruments, measurement of phosphorylated and total proteins on the same bead. And so my part today is to introduce you to Luminex's newest instrument for bead-based multiplexing, the xMAP INTELLIFLEX DR-SE. But before we begin, let's talk a little bit about the whole xMAP instrument history with Luminex. In 1995, the company was founded, and at that time we had a 64-plex of different color-coded microspheres and a software, but then you would have to run your assay on a flow cytometer. So very shortly, we determined that it would be best if we could design an instrument that was specifically optimized for running our beads. And we came out with the Luminex 100 in 1995. After a few years, though, we then upgraded and launched the Luminex 200 instrument. So this was an instrument that was very similar to the Luminex 100, but it had several hardware improvements based on feedback that we received from customers and partners. Then in 2009, we came out with the FlexMap 3D instrument. So this instrument was designed to meet the needs of the higher plex, higher throughput laboratories. Just a year later, we launched the MagPix, and this was really intended to meet the needs of the lower throughput, lower plex laboratories. And this was our first instrument that was based on bead imaging and not a flow cytometry-based instrument. But you can see here there was quite a gap until in 2021, we launched the xMAP INTELLIFLEX RUO instrument. Let's have a little bit of an overview of the Luminex flow analyzers. So the Luminex 200, as I said, was launched in 2005. It is a flow-based instrument. It can read up to 100 different bead regions per well. It analyzes 96 well plates, and it can complete 96 wells in about 45 minutes. And this means that you can get up to 12,800 test results per hour. The FlexMap 3D, the high throughput instrument that we launched in 2009, is also flow-based, but it can read up to 500 bead regions per well. And because of its dual sample fluidics pathway, it can analyze a 96 well plate in just 20 minutes. It's also compatible with 384 well plates, and it can read a 384 well plate in about 75 minutes. So this means that you can run up to 144,000 tests per hour in the 96 well format, or 153,600 tests per hour in the 384 well format. Now, so this sets the stage for now me introducing you to the xMAP INTELLIFLEX. So the xMAP INTELLIFLEX is that same versatile multiplexing platform that you've come to know and trust, but it's got some enhanced features to give an overall improved user experience. So if we look at the xMAP INTELLIFLEX RUO, this has a multiplex capacity of 500 and an improved dynamic range. It's now possible to get out to 5.5 logs in the reporter 1 channel. It has the 96 and 384 well format, but in this case, with this instrument, the PC and the monitor are integrated into the instrument, so it has a smaller footprint than the previous instruments. The read time is the same with a 96 well plate in 20 minutes or a 384 well plate in 75 minutes. And it's also got a modular design with fixed laser mounts. And then in the front, you can see that there's a wide, clear sliding door to make it really easy to access that sample probe to do your probe height adjustment. But what we're really going to be focused on today is the xMAP INTELLIFLEX DR-SE model. DR-SE stands for dual reporter side eject. And so you get all of this that I just described for INTELLIFLEX, but in addition, it's got the dual reporter configuration. Yes, we added another laser to provide a second reporter channel, RP2. And in this channel, you get 4.5 logs of dynamic range. And then as you can see in the bottom right-hand image, there's a side eject microplate carrier, which makes it really easy to connect this system to automation and liquid handling systems without having to turn the instrument, onto its side or backwards. So we're really excited about how we have modernized the Luminex flow analyzers. So let's dig into the INTELLIFLEX in a little bit more detail. So as you can see here, showing the dimensions front to back, side to side, and top to bottom, it's got a reduced footprint compared to both the Luminex 200 and the FlexMap 3D instrument. And this is because it's got this integrated PC and touch screen. I should mention, though, that for those of you who still like your keyboard and mouse, there is a port that you can hook those up if you prefer. The other neat thing about this system is the modular design. Inside, there's a plate module, a fluidics module, and an optics module. So this makes it really easy for our field service guys to get in there to do maintenance and upgrades and repairs and things of that nature. So speaking of the optics module, let's now look at the optics. So on the left-hand side, we're just showing the basic layout for the single reporter system. So here we have the red laser that excites and in the internal dyes inside the microspheres, and then the green laser, which excites the surface orange fluorescence to measure what is bound to the surface of the bead. And so on the left-hand side, where we're looking at the classification, we have our 3 avalanche photodiodes that measure the internal dyes inside of the bead and determine which bead set is being read. And then the DD, or doublet discriminator, helps us distinguish what is a single bead from clumps of beads or aggregates or something smaller than a bead. Then the green laser, which measures our reporter fluorescence, sends that signal to a photomultiplier tube, or PMT. And in addition, you see there's an EDR detector, which stands for extended dynamic range. And this actually allows us to measure the reporter signal beyond the saturation point of the PMT, and that's what gives you your 5.5 logs of dynamic range. And so the single reporter system has that standard 532 nanometer green laser. But now if we look at the right, we can see the xMAP INTELLIFLEX DR-SE model. And you see here we have added an additional laser to provide that second reporter channel. The classification is done exactly the same. The red laser excites the internal dyes and measures which bead set is being analyzed. The green laser measures the surface orange fluorescence for the RP1 PMT. And then the violet laser, which excites at 405, it measures blue fluorescence, and that signal is sent to a second photomultiplier tube, the reporter 2 channel, or RP2. And so the blue dyes that can be measured are in the 421 to 436-ish range there. And so this is how we were able to achieve not only a 500 plex of beads, but now 2 different readouts per each one of those beads. So now that we understand how the dual reporter works, let's talk about performance. Here we have some data that was provided to us by our partner at Thermo Fisher Scientific, and we definitely appreciate that. What they did here was they ran their cytokine chemokine growth factor 45-plex human procardioplex panel to look at the results with some samples and, of course, standard curves for various cytokines. Here we're just showing the data for human IL-8. And on the left, we have those standard curves. And so what you can see here is what we call backward compatibility. So whether you ran the experiment on a Luminex 200 or a FlexMap 3D, you can get virtually identical results on the INTELLIFLEX by selecting the appropriate operating mode. So here we have data from run on the Luminex 200 in blue, and together with it, the data from INTELLIFLEX run in the Luminex 200 mode. So this is shown in red, and you can see that those cytokine standard curves are laying right on top of each other. Similarly, if you ran your experiment on a FlexMap 3D in green, but then you ran it on the INTELLIFLEX in the FlexMap 3D mode, shown in purple, those curves are also virtually identical. And so what this means is if you are used to running your assays on a particular instrument and setting, you can achieve the same results on the INTELLIFLEX just by selecting that operating mode. And so you should be able to take your assays and port them right over onto the INTELLIFLEX seamlessly. On the right, we're showing some results for various samples that were run. And again, you can see that whether you ran it on a Luminex 200 in blue or in the INTELLIFLEX Luminex 200 mode in red, the FlexMap 3D in green, or the FlexMap 3D mode on INTELLIFLEX in purple, those points are clustering all right next to each other, exactly how you would expect them to, with approximately the same MFI values and virtually identical picograms per mil. Okay, so now that we see that the performance is tight, let's talk about applications. So here is a very simple single reporter example. This is to sort of get us thinking about how the beads are measured in the various instruments. And so here we're showing just a very simple serological immunoassay, an indirect immunoassay format where we have an antigen coupled to the bead, and then we're trying to measure specific antibody in the sample. So now something that we maybe don't think about so much is all of the antibodies that are specific to that antigen are going to bind to the bead, but we can only measure one of them at a time. So in this case, in our first well 1, if we want to measure IgG, then we put in an anti-IgG detection antibody labeled with phycoerythrin, and then we can measure the specific IgG that has bound to that microsphere set. Now, what if we also wanted to measure IgA? It's there, we just have to have a way to detect it. Well, in the single reporter instrument, you have to run a second well. So here in well 2, we have an anti-IgA detection antibody that's also labeled with phycoerythrin. So on the single reporter instruments, this is how we would do isotype measurements. But now with INTELLIFLEX DR-SE, we can put both detection antibodies in together at the same time with the appropriate dyes. So here we're showing the anti-IgG detection antibody with the phycoerythrin attached. And then to measure IgA, we have an anti-IgA detection antibody that's labeled with one of the blue dyes that can be measured by the violet laser. And so you can see in the little cartoon on the left, the bead is going through, it gets classified with the red laser, it gets excited with the green laser to measure the phycoerythrin, and then excited with the violet laser to measure the brilliant violet or super bright 436, whichever dye you decided to use for that second channel. So in this way, you basically double your data. You can measure 2 parameters of your assay at the same time on the same bead in the same well. So now that we kind of have that idea in our head, let's look at some real data that was generated with the dual reporter instrument. In this case, we used our Luminex xMAP SARS-CoV-2 multi-antigen IgG assay that received emergency use authorization from the FDA. But what we did here was we swapped out different detection antibodies with different dyes so that we could test the dual reporter feature and measure IgG and IgM at the same time. So on the left, we're just showing what we measured if we only had the IgG PE labeled detection antibody present in the well, but we are measuring it in dual reporter mode. And so you can see here we have a nice dose response increase in signal in PE as the serum is more concentrated. And we have background measurements for the RP2 channel because there was no reporter there to be measured. In the second panel, B, we left out the IgG-PE labeled antibody that goes in RP1, and we only included the super bright labeled IgM antibody, which is measured in reporter 2. So you also see that you have a nice dose response rise in signal with the more concentrated the sample, and then the signal in the phycoerythrin channel is background levels. I know it looks like it's kind of creeping up there, but remember this is a log scale, and so those are still, just a few tens of MFI, so definitely still at background levels. But what's more important is if you look at panel C, this is when we measured the 2 antibody isotypes at the same time with both detection antibodies present in the dual reporter mode. And this is shown by the solid lines. If we then take the individual reporter data that we measured previously in the dotted lines and plot them all together on the same chart, you can see that the results are virtually identical and those curves are laying right on top of each other. So what was very important about this experiment is that it proved that you could use both reporters together at the same time, and there's no interference with each of the reporter lasers or the reporter dyes, which of course you wouldn't expect because they're completely different wavelengths, but we still had to prove it. And so this experiment gave us some confidence that the dual reporter was working exactly the way it was intended to. So next, we wanted to look at some data with some real samples. And so, this was happening right at the height of the COVID-19 pandemic. And what we see on the left was we took measurements from real samples from patients in looking at the time after vaccination. So you can see that after about a week after vaccination, you get a rise in signal for both RBD-specific IgG and RBD-specific IgA. And so the IgG was measured with phycoerythrin, the IgA was measured with brilliant violet. And so this was a nice, robust response just a week or so after vaccination. And then you can see with the black arrow at about 21 days is when everyone received their boost. And then you can see the signal shot up again. The IgG levels stayed very constant out through the about 45 days of measurement, whereas the IgA started to dwindle off after about a month and a half. In the center panel, we were looking at different patients. And so here in the pre-COVID era, you would not expect to see any signal for any of the antigens. So we're looking at the RBD antigen, which came with the vaccine, and then also the N antigen, which was part of the virus, but not present in a vaccine. And so the blue dots show you that prior to the COVID-19 pandemic and outbreak, nobody had seen this virus before, so nobody had any antibodies. Then if you look at the black dots, you're seeing a high signal against the RBD, so IgG raised to the receptor binding domain of the virus, but no signal on the N. So these are patients that had received the vaccine but had never been infected with the virus. Versus the red dots, these are patients that were previously infected with the virus, so they had high titers to both the RBD antigen as well as the N antigen. And so I'm guessing that now, here we are a few years later, all of us would probably be up in that red cluster for the most part. Now, on the far right panel, this is something that we did to see how comparable the results on the INTELLIFLEX using Brilliant Violet in the second reporter channel were compared to the signal for phycoerythrin in the previous instruments. And so you can see here that when we were measuring IgA with phycoerythrin on the MagPix, the Luminex 200, or the FlexMap 3D in red, blue, and black, we got very similar signals and very comparable results compared to the signal that we measured on the INTELLIFLEX DR-SE using a Brilliant Violet labeled detection antibody. And you can see we've got, slopes near 1, R-squared's nearly perfect. I should mention, though, that it does not mean that 1 MFI of phycoerythrin equals 1 MFI of Brilliant Violet, but we just got very similar MFI values in this situation with these dyes and these samples. So it's kind of, you can start to think about what you might be able to do with this new dual reporter capacity. And already we see a number of publications coming out using this new feature on this new instrument. And so here, I wanted to show you a few papers that have been published. Start at the beginning. These first few papers, Angeloni, Fayed, and Cameron, these are all dual reporter assays that were generated to SARS-CoV-2. So looking at different antibody isotypes at the same time, in the same well, on the same bead, using the INTELLIFLEX in dual reporter mode. And so each of these were actually multi-antigen immunoassays developed by these different investigators. And so very similar to the data that I showed you previously using R-KIT. But then the next paper by Gao et al. Is looking at an epitope peptide vaccine to measure the anti-tumor immune response in mice models and then subsequently human models as a blockade checkpoint inhibitor PDL1 PDL blockade assay. So in this assay, they actually used the dual reporter in a competitive immunoassay format to look at the blocking of binding with this antibodies raised to this vaccine. So that's a really interesting and kind of novel application. And then moving on to the next set of papers, some other serological assays, but for other diseases. So we have from Herring et al. A Borrelia multiplex, simultaneously measuring IgG and IgM to a multiplex assay looking at antigens for different Borrelia species. And then Shiver et al., the development of a duplex serological multiplexed assay for measuring IgA and IgG to Epstein-Barr virus simultaneously in nasopharyngeal carcinoma patients. And then the next two papers are actually from our previous authors, but in this case, they each published their experimental method in the Journal of Visualized Experiments. And so in this case, you can actually go and pull these papers and get their exact experimental protocol. And then subsequently, the Journal of Visualized Experiments publishes a video of the authors performing the assay in their laboratory. And then the last paper, Sahi, is also looking at profiling surface protein epitopes on viral particles using the dual reporter strategy from the INTELLIFLEX DR-SE model. And so that one just came out earlier this year. So already 9 papers have been published just since we launched the instrument in 2021. And that's quite a contrast to when the whole company first started. In the first 4 years that the Luminex 100 was out, there were only about 4 or 5 papers. So this sort of tells me that the dual reporter is already taking off, and I can't wait to see all of the interesting things that everybody uses it for. So that brings me to this little cartoon here, thinking about dual reporter applications. So now you have one target that's on the bead, but you get 2 different readouts. So as I mentioned before, you're doubling your data. And think about all the different things that you can do. You can look at isotyping, just like we did, measuring two different isotypes to the same antigen. You can look at drug and ligand interactions, looking at free and bound drug forms, bispecific molecules, fusion proteins, as well as protein-protein interactions, just by coming up with the right combination of reporter detection molecules with each of the two reporters. And so, of course, the one that I saved for last is the first one that I think of, which is looking at post-translational modification. I mean, this is an ideal setup for looking at measuring total protein as well as phosphorylated protein together at the same time in the same well. So what the dual reporter function does for you is it takes out yet another variable that could occur if you're measuring in two different wells. And so with that, I am finished introducing you to our instrument today, and I can't wait to hear what our next speaker has to tell you about phosphorylation.
Thank you very much for the introduction and welcome again to this webinar. In the second part, it's my pleasure to introduce our new ProCartoplex dual reporter assays as the first application for the Luminex INTELLIFLEX DR-SE instrument that have been specifically designed to simultaneously interrogate key signaling molecules and their phosphorylation state to help gain deeper insights into the molecular mechanisms of cell signaling pathways and to advance our understanding of physiological processes, but also related pathological conditions. So the complexity of cell signaling pathways is outlined here already in a more simplified scheme and exemplified by AKT, also known as protein kinase B, which is one of the most extensively studied kinases in different key research areas, including drug discovery and development, that acts as a master switch within a broad and dynamic intracellular signaling network that ultimately enables cells to sense and respond to environmental cues. So this signaling cascade is typically activated via insulin or growth factors that promote the phosphorylation, so the addition of phosphate groups to corresponding receptor tyrosine kinases that subsequently activate further downstream targets. And as illustrated in more detail on the right, upon phosphorylation of two key residues, AKT further propagates the signal transduction through the activation or inhibition of multifunctional nodes and other downstream effectors that greatly expand the functional spectrum of this kinase with some overlaps and also crosstalks with other major signaling pathways to finally coordinate various cellular processes as shown here, including cell growth and proliferation, metabolism and protein synthesis, or processes related to the immunity. However, dysregulation of these networks also underlies a variety of pathological disorders associated with metastasis, inflammation, or drug resistance, which is also an active area of research to discover new potential components for targeted treatments. So given the range of different AKT substrates and the complexity of feedback loops and cross-regulation involving also other interacting partners, understanding molecular mechanisms of cell signaling still pose a challenge. To address this, various methods and platforms are available to meet the diverse needs in cell signaling research as summarized here, from mass spectrometry supporting discovery proteomics and commonly used techniques for the verification of targets, including Western blot, flow cytometry, imaging or high content analysis, to the validation of individual specific analytes at the single target level, such as by ELISA. In this context, ProCartoplex immunoassays, and in particular the new dual reporter assays, are valuable high-throughput tools to complement these and other technologies, enabling an even more refined and efficient quantitation of multiple targets, including post-translational modifications of the same proteins in a multiplex format on the Luminex system. This highlights key features of ProCartoplex multiplex immunoassays that allow for the simultaneous analysis of up to 80 protein targets in a single well of a 96 or 384 well plate from a variety of sample types, including also cell lysates, with just low sample volume requirements, and also provide the highest flexibility to mix and match specific analytes of interest that can be selected from a broad list of available targets to create custom research panels appropriate to the respective research needs. So applying the multi-analyte profiling technology, procartoplex immunoassays enable protein target identification via binding with a highly specific antibody pair in an ELISA-like workflow and detection via fluorescent reporter molecules in a Luminex instrument with a time to result of only 4 hours. The figures on the right further illustrate in more detail how it works. So basically, the technology relies on sets of color-coded and spectrally distinct magnetic beads that can be classified by the red laser in any Luminex instrument. And each bead set is coded with a capture antibody that specifically binds to a target protein of interest. So adding a sample to a mix of capture beads finally allows the analyte-specific binding of multiple targets from a single well in parallel that can be detected following incubation with a mix of target-specific biotinylated antibodies in combination with fluorescent reporter molecules. So in a typical procartoplex assay, streptavidin phycoerythrin finally allows for the quantitation of individual analytes bound on each bead via the green laser and first reporter channel, while the new procartoplex dual reporter assay format enables the quantitative readout for a second parameter on the same bead by introducing brilliant violet 421 as a new dye in the violet spectrum that is compatible with the additional laser and second reporter channel of the Luminex INTELLIFLEX DR-SE instrument. Taking a closer look at the design of the different procartoplex assay formats and the new capabilities of procartoplex dual reporter assays demonstrated here in more detail, as shown on the left, 2 new procartoplex panels are now available for cell signaling research that enable the measurement of either 8 total or 8 phosphorylated proteins separately by utilizing beads coupled with target-specific pan-capture antibodies that bind to signaling proteins independent of the phosphorylation state, and either pan or phosphor-specific detection antibodies in the procartoplex total or respective phosphor-specific assays, both using streptavidin phycoerythrin as a fluorescent dye for the readout via the green laser and first reporter channel, which is suitable for all Luminex instruments. The design of the new procartoplex dual reporter assays is shown on the right. So by introducing a new fluorescent dye for the phosphor-specific detectors and second reporter channel in the INTELLIFLEX DR-SE instrument, as well as a new TAC anti-TAC system next to biotin and streptavidin for signal amplification and detection of total proteins via phycoerythrin in the first reporter channel, the new procartoplex dual reporter assays allow for the simultaneous quantitation of up to 8 total signaling proteins and their phosphorylated fractions in one well. As mentioned, various techniques are employed in cell signaling research that allow to choose the appropriate and most suitable method to help best answer your research question. And this table summarizes key features and differences between Western blot, ELISA, and procartoplex multiplex assays. And I'd like to point out just a few. So while Western blot particularly facilitates the visual identification of proteins and also provides information on the protein molecular weight, ELISA and procartoplex assays enable a specific and sensitive protein quantification via calibration curve, with the particular benefit of procartoplex assays allowing for high throughput profiling of larger sets of analytes in a single sample run in parallel with less sample input, while the dual reporter assays, in addition to the multiplexing capabilities, enable the simultaneous quantitation of 2 parameters per beat, so the phosphorylated and non-phosphorylated state of the same protein for multiple analytes. Before I'll share data on the side-by-side performance comparison between these platforms for selected representative analytes, I'd like to further highlight a key feature of the ProCartoplex assays, including the new dual reporter assay format, which is a quantitative readout that is enabled for all signaling molecules, as shown here, via calibration curves using phosphorylated proteins. And these have also been used to further verify the phosphospecificity, as shown in the graphs on the right, for AKT total and phosphorylated AKT assays. So while a treatment of post-translationally modified recombinant proteins with a lambda protein phosphatase resulted in dephosphorylation and concomitant loss in the signal of the calibration curve in the phosphospecific AKT assay, indicated here in green, compared to that of the untreated recombinant protein shown in blue, the signal of the treated and the phosphorylated recombinant protein remained unchanged in the AKT total assay, as shown on the left. As highlighted here and outlined previously, AKT is activated by insulin and various growth factors and is known to function in a pathway that is sensitive to wartmanin, an inhibitor of the phosphoinositide 3 kinase activity upstream of AKT. So using ProCartoplex assays, as shown in the bar chart on the right, analysis of the levels of AKT total, shown here in blue, and phosphorylated AKT, shown in red, in lysates of Jurkat T cells treated with different concentrations of mortmanin, clearly demonstrates a dose-dependent decrease and inhibition of AKT phosphorylation with increasing concentrations of this inhibitor, as expected, which further verifies the performance and underscores the value of ProCartoplex assays for cell signaling research studies. Moreover, side-by-side analysis of the concentrations, as shown here for 3 selected phosphorylated target analytes measured in various cell lysates by in Vitrogen ELISA and ProCartoplex assays, revealed that ProCartoplex assays achieved quantitative results consistent to the ELISA kits, with the additional benefits of multiplexing, including a reduced sample volume that is needed. Performance comparison of protein analysis between ProCartoplex assays, as shown on the left, and Western blot, shown on the right, furthermore demonstrates high concordance between the two systems. So ProCartoplex and Western blot data show similar levels of the phosphorylated proline-rich AKT substrate plus 40 in murine fibroblast cell lysates that were found to be considerably induced in response to treatment with a platelet-derived growth factor in comparison to the uninduced control, and largely inhibited by addition of wartmanin that blocks the activity of upstream regulators in this pathway, as shown previously. Further side-by-side comparison of the sensitivity between ProCartoplex assays and Western blot, as shown here, using the same amount of a 2-fold serially diluted recombinant protein and the same detection antibody specific for phosphorylated cyclic AMP responsive element binding protein in both systems, revealed a similar pattern and trend with a slightly higher sensitivity of ProCartoplex assays as compared to the lowest concentration that could be detected and clearly differentiated from the background by Western blot. Similarly, when the levels of the phosphorylated CRAB protein were determined in serially diluted, more complex Jurkat cell lysates, as shown here, ProCartoplex and Western blot analysis revealed comparable sensitivity and performance. Now, taking a closer look at the performance across the different ProCartoplex assay formats, side-by-side analysis of ProCartoplex simplex kits and ProCartoplex AKT pathway 8-plex panels, as shown here for AKT total and phosphorylated AKT, further confirms the scalability of procartoplex assays, showing a reproducible performance regardless of the plex size, which makes it easy to switch between simplex kits and multiplex panels. Further performance comparison between the procartoplex AKT pathway 8-plex panel for either total or phospho and respective dual reporter panels, as shown here for mTOR total and phosphorylated mTOR, demonstrate a strong correlation with high sample correlation coefficients and consistent reproducible results also between the two formats. To support and further enhance also data analysis of the new dual reporter assay format, the Procartoplex Analysis app has now been updated and is available as a free software via Connect, the Thermo Fisher Scientific cloud-based platform. So besides established 4PL and 5PL curve optimization, heat map analysis, or group-wise comparisons, new features will now also allow the calculation of ratios between total and phosphorylated protein and fold change versus control, as demonstrated here by the data obtained for the glycogen synthase kinase 3 beta, as shown in the bar charts. So as a key component of the cellular responses to growth factors and insulin in the AKT pathway, the levels and concentrations of phosphorylated GSK3 beta were found to be considerably increased in lysates of HepG2 cells treated with IGF-1 or insulin, whereas the levels of the total protein remained largely unchanged, resulting in a respective calculated ratio, as demonstrated here, and a twofold change in the concentration of the phosphorylated target in comparison to the uninduced control. So to conclude, besides the new procartoplex panels that are now available for the interrogation of either total or phosphorylated key protein targets on any Luminex instrument, the new procartoplex dual reporter assays now provide a unique tool on the INTELLIFLEX DR-SE system for the simultaneous quantification and differentiation of multiple total and phosphorylated signaling molecules in a single well, including even low abundant proteins, with comparable results to ELISA and Western blot analysis, however with higher throughput and less sample input that is required in a multiplex format. They are therefore ideal tools to support cell signaling pathway analysis and targeted drug discovery and development studies across key research areas. And with that, I'm at the end of my presentation, and thank you again for your interest in this webinar.
Thank you so much to Dr. Dunbar and to Dr. Badrnya for their informative presentations. And thank you, Dr. Badrnya, for coming back to join us for the live Q&A. Now, we have a lot of questions that have already come in. And to our audience, if you haven't already submitted any questions, please do so now. Just type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the right now, actually. So let's dive right in. Being that we have so many questions, I'm just going to start from the top, work my way down, and let's begin with this one. So, are you working on more assays for the dual reporter instrument?
That's a good question. Yes, indeed. So you can expect more assays to come later this year for analytes that are relevant, especially in this specific research field and key area, leveraging the functionality of the dual reporter is in the INTELLIFLEX DR-SE instrument. And in this regard, I'd like to also point out that for the procartoplex AKT pathway panels that are now available, there's also a matched complementary quantigeneplex panel for gene expression profiling of relevant target molecules on the Luminex system that might also help to gain even more comprehensive insights from a lyzed sample and also facilitate and interrogate more complex signaling networks with a multi-omics approach without switching the instrument platform. And if there is any other target of interest or application where you want to take advantage of the dual reporter system in the INTELLIFLEX DR-SE instrument, please reach out to us. So if you cannot currently find it, perhaps our free consultancy services and offering for custom assay development is a good option and solution to your respective needs.
Thank you. Now, staying on the topic of Western blot, is the limit of detection of these signaling kits lower compared to Western blot?
That's a good question. So in general, the sensitivity depends and varies depending on different factors, the target, the antibodies that are used. But as we have demonstrated by examples in the presentation and side-by-side comparison using the same phosphorylation specific detection antibody in both systems, it revealed that in some cases, there might be a slightly higher sensitivity. Overall, it was found to be at least comparable to the Western blot.
Now, do procartiplex assays have a shorter protocol than Western blot?
So this really depends on the system that is used, right? However, as outlined previously, procartiplex assays have a very workflow that is straightforward, enabling the simultaneous quantitative detection of larger sets, as well as now up to 2 parameters per bead using the dual reporter assay format. And taking only 4 hours from sample to answer, and typically also less sample input that is required as compared to a conventional Western blot protocol that involves many manual steps.
Thank you. Now, this question has come in a few times. Can I use these assays with mouse samples?
So as the sequence motifs of several signaling molecules are highly conserved across different species, mouse samples are suitable, and we have demonstrated in the presentation here for plus 40 levels, for example, that have been verified in a mouse embryonic fibroblast cell line and lysates of this. So yes.
Excellent. Now, speaking of lysates, is a specific lysate preparation needed?
So for the preparation of lysates, we recommend a protocol and starting concentrations of around 1 to 10 to the 7 cells per ml, and the use of 2 different lysis buffers as stated in the user guide for these assays. So either procartiplex cell lysis buffer or cell extraction buffer complemented ideally with a protease inhibitor cocktail. However, depending really on the cell system you are using, other buffers or other protocols can also be suitable as well.
Thank you. Our next question, is it also possible to turn off the second laser for experiments where I don't need it in the INTELLIFLEX DR-SE system?
So there's not necessarily a need to turn off or turn on specifically a laser, but you have the possibility and the option to choose between different operational modes for either just the single first laser and reporter channel or a dual reporter mode. So this is recommended to select whenever you are running a dual reporter assay.
Very good. Now, we have quite a few questions coming in asking about this one. I wonder if I understand well, the MagPicks do not suit for this modality of assays. Do you need the INTELLIFLEX instrument for this?
So actually, the dual reporter assays, as mentioned, have been specifically designed for the additional laser and second reporter channel that in the INTELLIFLEX DR-SE instrument. However, the Procardiplex assays for cell signaling research are available in different formats. So they include also two panels that enable the simultaneous measurement of either total or phosphorylated proteins that are suitable with any of the Luminex instruments, including also the MacPix or a Luminex 200 instrument. So,
Thank you. Now, we're also having some questions coming in on extracellular vesicles. Do you have experience with extracellular vesicles?
we do have experience with extracellular vesicles. It's they are acknowledged as a rich source of biomarkers and especially also the protein cargo, and are an exciting new sample type and sample matrix that has been also verified on the Procardiplex platform. Right.
Thank you. Now, how much sample do I need for this assay?
So to simultaneously measure up to 8 total or phosphorylated targets, or even 16 parameters when using the dual reporter assays in a single well, you need 25 microliter of a pre-diluted sample. We recommend, as indicated also in the user guide, sample pre-dilution of 1 to 5 in the respective assay buffer that is provided with these ready-to-use pre-configured panels. However, higher dilution may be necessary depending on the cell model or the type of cell lysates you are using. So overall, in total, the sample input volume is fairly low.
We have questions coming in. Let's see, so many. I do want to remind our audience, though, any questions that we don't have time to answer today, and additionally those that are submitted during the on-demand period, they will be addressed via the contact information you provided at the time of registration, but we're going to try to get in as many more as we can in the time that we have left. So let's go with the INTELLIFLEX. Can it give similar MFI values for both Luminex 200 and FlexMAP 3D? And then how about the MagPix?
Yeah, so as pointed out and demonstrated in Sherry Dunbar's presentation, there is really consistency across the different instruments, including also the MacPix instrument, right? It was demonstrated for FlexMAP 3D, Luminex 200, but this holds also true for all the other instruments.
Excellent. And we have had quite a few questions coming in asking for pricing and for demos for this product. We will be having somebody reach out to you to speak with you directly, as well as set up demos. So if you are interested in any of that information, please just drop in some information about yourself in the Q&A box, and one of our team members will reach out to you. So I think we have time for one more question. I already have the Luminex 200. Can I run your dual reporter assay on it?
So, well, as I've pointed out, I think in the previous question, so the Procardiplex assays for the for cell signaling research are now available in different formats, and of course you can use the AKT pathway 8-plex panel for either total or phosphorylated proteins on any of the Luminex instruments, including the Luminex 200. That's possible.
Excellent. Dr. Badrnya, thank you so much for your time today and for joining us for the Q&A. And I also want to thank Dr. Dunbar as well. We appreciate you sharing your knowledge and your expertise, both of you, in advancing research through the utilization of cutting-edge technology and innovative assays. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Dr. Badrnya, do you have any final comments for our audience?
So It was a pleasure. Thank you for your interest in this webinar. And yeah, as a partner of Luminex, we are pleased to provide really the first application and this assay solution that will enable you to advance cell signaling analysis and really accelerate also targeted drug discovery studies across the different key research fields. Thank you very much.
Thank you. And I do want to thank our audience also for joining us today and for their interesting questions. We did have a lot of questions that came in that we were unable to answer. So I do want to remind you that those will be addressed by our speakers via the contact information you provided at the time of registration. Now, today's webcast can be viewed on demand, and LabRoots will send you an email when it's available for replay. We encourage you to share that email with your friends and colleagues who may have missed today's live event. To all our friends out there, thank you for joining us. Have a great day, everyone. Bye-bye.
Thank you, everyone.